All right. Thank you again for all of you who have joined today's session and wanted to learn about Cheminformatics in biomedical drug discovery. So for today's session, we discussed and we thought that it will be nice to discuss some of the critical opportunities and challenges that are being faced by the industry, faced by research labs, and we wanted to discuss about the overview of how this entire modern drug discovery pipeline is now functioning and it's producing results in many places. And many of you are excited for the same reasons to be able to explore more of this and to be able to learn and apply to some of your own research. So as we go on for the session, as we discuss different agendas for today's meeting, I'd like to request Sonalika to maybe prepare a project proposal form and share it with me. Sonalika, if you can do that. So towards the end of the session, I would want to discuss the project proposal and how one can start working on a research idea already, starting with as the program starts in a week's time. So as we have been discussing that there are certain agendas that we would want to cover. One of those agendas is precision medicine. So when we are discussing about biomedical drug discovery, when we are discussing about the use of cheminformatics in biomedical drug discovery, we are talking about a specific specialization that involves structural biology, that involves understanding of how to analyze three-dimensional structures in three-dimensional space, and how to devise strategies as well. So I mean, to think about that, there are other two aspects that we would be discussing. Those are drug designing and drug repurposing, which are used quite often in even in industry as well as in academia. And during the presentation and during the session, I'll be happy to answer questions. So please put your questions in the chat. So in this training program, what we have discussed so far is about the different aspects of biomedical drug discovery. But in the training program, we are going to follow a curriculum, and that curriculum would include specific softwares and tools which are open source, and we will be able to share with you how to install those softwares and how to use them on the cloud, and what are the associated resources that you can utilize to make the most out of this training program that is upcoming. So we have planned a very detailed curriculum that I'll be showcasing and I'll be going through. But before that, I wanted to start with the fact that what we are doing and why we are doing this and why we should learn about all of this. So let's start with the fact that how the modern biomedical drug discovery pipeline works and what are the different components, and how can this be an entire story, like a complete story with all the where we know all the pieces. Because what has happened and what we know for sure is that for many drugs that we've been using so frequently, we don't know the mechanism of action. So having a complete knowledge of mechanism of action is something that the modern biomedical drug discovery is utilizing that is being utilized by pharma, computational biology, or bioinformatics company that are providing services, as well as research labs and researchers. So how do we incorporate that all sort of information into the model so that it makes sense, so that it relates really close to what is happening biologically or what is happening in a real life situation. So I mean, in modern biomedical drug discovery pipeline, there are certain aspects that we have covered in our OmicsLogic portal. And to those participants who will be joining this program, obviously connecting those dots and then figuring out maybe target protein or a proposal would make total sense. So as we were discussing precision medicine, so precision medicine initiating from clinical and molecular pharmacology introduces a new era in healthcare, right? The entire purpose of this is that it aims to identify, and predict the optimum treatment for a patient or a cohort. Now, evidence-based medicine integrates information from basic in vivo observational studies, and then clinical trials were done to do meta-analysis data for clinical considerations. So hence, it can be stated that this entire, like, often we see that population averages, but population averages the forest but misses the tree. So the trees are here individual patients, right? Where the utilization of precision medicine may not see the forest, but for the trees, and that is quite relevant and important, that we have realized that all of us are having a different genetic makeup, which makes us different from each other. We react to different medicines differently. So all that information can be, modeled together, so that we can reach to a place where we can give recommendations based on the personalized way of reporting. So now the state of art tools for modeling and simulations in pharmacology, they try to extrapolate knowledge gained through this experimental process or procedures with either a top-down approach or a bottom-up approach. So this modeling and simulation often, it what it does is that it tries to connect the dots and reveals the bigger picture. So the bigger picture is the population or the forest. So considering what kind of individuals or trees are in there, we have to understand, and we have to figure out at an atomic level, and which is very fascinating and really nice to know that from a sequence level, we are looking at the functional level, and then we are looking at the mechanistic level by looking at the chemistry that is going on within the molecules. So in this program, there are some components that is really exciting for me, and I guess it would be exciting for all of you as well. So we would want to discuss this, that how research allows us to utilize experimental or clinical data through this molecule modeling and simulation of biomedical tools. And this will help, and this is helping for sure in disease progression dynamics, in machine learning and relevant bioengineering approaches for either drug response prediction, where, like, including drug interaction data is also included, then adverse drug reactions data is also included, and that all of this is leading to drug repurposing or drug discovery. Now, we also want to understand that what different this slide itself. It has a huge number of information, but at the same time, it also tell us about the pathway, like what cellular omics can give us, what microbiome and virome information can give us, and what clinical and phenotypic data can give us. And when we combine all of this, what is a system biology approach, and how we can then discuss about the strategy. The strategy is to find a solution, and the solution comes from chemistry. So how do we use those drugs, those maybe biologics, or based on the data that we are getting? So this various types of omics data that is described in detail intercellular and intracellular processes, which are really associated with common diseases. And these data sets can be analyzed to understand biology behind host factors, and as well as like a microbiome composition in our bodies and the environment we live in. So to drive infections and to cause deterioration of health, these also have an effect on destabilizing the immune system, and all of this led to clinical manifestation of a disease. So it would be nice to understand this from a perspective of an example so let's try out an example. So the way in which bacteria infect cells are important for understanding the host pathogen interaction. Agreed? So if it is true, then the knowledge also opens up a world of practical applications. So what are the applications? So for example, it can support the design of new antibacterial or vaccines in In case of when we are discussing about healthcare, these are the different applications. When we are discussing about the soil or marine bacteria, it can help in creating strategies for rebalancing natural ecosystems. So there is a huge application. So one can use machine learning to explore how bacteria interacts with the environment by analyzing those. So there is this case study, and there's a recently published article in Nature that shows that analyzing data from Uniprot database in itself and using the protein prediction software that recently came out, AlphaFold, uh, one can bridge, like fill this gap and knowledge in this area. So there is this study where fibrillar adhesins, these are the proteins that are located on bacterial cell surface, and which mediate interactions with the environment, for example, host cells or the bacteria or the other bacteria. So there was a study on this. So these proteins are usually have a stalk which helps them cross the bacterial cell surface and be projected closer to their targets. So regardless of what they are trying to bind to, they will be this is the mechanism through which they work. So the study analyzed the data from a couple of species, and then the researchers were able to identify more than 6,500 of such fibrillar adhesins that were not seen before. And this was done through the sequence analysis. So Uniprot is a sequence database, and I think many of you know about Uniprot very well. And then we will be discussing in this session also AlphaFold and DeepMind's this innovation, which helped in predicting the structure of those fibrillar adhesins. And then we got to know about from this analysis a new world, perhaps, that is helping in the process of binding, which is so essential for bacteria. So this work actually goes beyond simple curiosity, because by understanding how bacteria interacts with biotin or abiotin surfaces, researchers can start to explore how drug can be used to target these interactions, or whether the bacteria living in our body or our environment can be changed. So this approach could allow researchers to improve the health of gut microbiomes, the bacterial community living in a patient's gut, for example. So these are some of the approaches that have been taken up by the new multi-omics way of seeing things, and then integrating it with the system biology approach, and then the computational solutions. So in this program, and we've been discussing this, that how the different elements are put together. So so when we are discussing about finding new targets, and when we are discussing about the data analysis part of bioinformatics, so you have heard about transcriptomics and like how gene expression patterns in association with clinical data help identify molecular biomarkers, which are used for diagnostics and for therapy selection. So new discoveries include a better understanding of these complex diseases at the subcellular level, as well as for target identification, for novel drug discovery, and even for drug repurposing. So all of this is helpful for us to towards target discovery, which is a very important step in biomedical drug discovery. Now, new target discovery. So target-based screens, as many of you are from maybe biological background and know about the target-based screens. So in wet lab or in when we are doing experiments, so target-based screens offer a reductionist approach to drug discovery, because generally it's employed in vitro biochemical assays to search through the libraries of small molecules. So they are based on developing assays to detect compounds that interact with a specific molecular entity, most often a protein, right? Which is known or hypothesized from a basic research to be involved in processes impaired in disease of interest. That's the drug discovery concept that many of us think it to be. So in that process, phenotypic screening, in contrast, employs a holistic approach, most often at the level of the cell. Sometimes you get that data also from cell, from tissue, or from organ, or even whole animals can be employed in some of those studies. So one of such example is that the personalized medicine example is trastuzumab, trastuzumab, which was a first-in-class immunotherapeutic targeting the HER2 tyrosine kinase receptor, and it's used to treat patients with HER2 overexpressed breast tumors. And we have a project on those, which project on this on Learn Portal, and also we cover it in our transcriptomics program. And but it is important that how all of this is connected. So there is a molecular approach that uses all of these technologies. I was discussing about and gave example for gene expression studies, where we discussed about a biomarker, and then the overexpressing HER2 breast tumors. And then there are genetic studies or genomic studies where we are looking at the panels, where we are looking at the mutations, the variants. So these are some approaches. Then there is a system approach that I've discussed. Then we have all the different validations that we can do utilizing bioinformatics. So this was a kind of an overview to what we are doing, why we are doing this, and how all of this is connected. Because on the portal, you will be able to find different omics technologies, and then why we are doing cheminformatics. So this is one of the major component of this entire drug discovery process. So I hope that all of this overview is kind of makes sense and is clear to you. If you have any questions, I'll be happy to answer. So next part of this presentation will include that what we are going to do in terms of the curriculum, how we are going to cover different aspects of what we have been discussing, and how all of this makes sense if you think about a research proposal or a research project that you might be thinking about or you are interested in the future to work on. So today we are meeting here and we are discussing about the upcoming fall program, which is Cheminformatics for Biomedical Drug Discovery. So this program is going to start on 23rd August and will run till October. And there are models where you can join and complete the program in either 45 days or 60 days or 90 days, based on the maybe you are interested to work further with us or work on a research proposal. All of this is possible within a larger time frame. And in this program, we will be covering through sessions, and there will be resources. So in this slide, I wanted to simplify to all of you who are here today, that how this is conceptualized and how the curriculum works, and what are the different components that one will be having access to be able to get some of the outcomes that we've been discussing for this. So we've been discussing about the different types of data, different types of approaches, and how structural biology approach is an important critical component of this. But you have like all the coursework associated with the other omics technologies that could be integrated into this process. But if you want to work on structure biology, cheminformatics, and you already have a defined idea or a research target, or sorry, target or a target in your mind, then I think you can start uh, from the uh, from the coursework that we are going to offer as a part of this program, which is structural biology and cheminformatics coursework, which kind of is developed for beginners. And this is especially to those who are either from high school or from undergraduate level and would want to start with a very basic understanding. So all of this OmicsLogic training involves a basic coursework, then an intermediate coursework, and then the research work. So for those who wants to get like an understanding of how the basics work, we will be sharing the resources with you. These are the courses or lessons that will be provided with every session throughout the training program, so that everyone who is from a different educational background or are from non-biology background could also work on this. So, as I was saying, that very basics to understanding of why we are doing this, where we can find information, and what kind of data is there. These all questions are also being answered. So, how protein acts, what is the future of structural biology, how NMR spectroscopy works, what is cryo EM, and what are the difference between like the when you compare structures, when you're thinking about conducting experiment, and in silico experiment on structural biology, drug discovery, drug designing, what are the things that you need to see? So there are some of the very basic important facts when you are obtaining data, when you're obtaining structural data. What are the parameters you can check to understand if the model that you're considering is going to give you the right answers or not. So those basic concepts are being discussed in these lessons, and we are adding more lessons in this series as well, which we will be sharing with the participants of the program throughout the training.